on computers, and that is required to do the sequencing and assembly as well, and the things that you should know about the genomes at the end. Okay, so let's start with the brief history of genome sequence assembly. So I'm pretty sure that you guys know a basic about genome sequencing techniques evolution. And I think some of the in previous lectures, some of the section is being covered. So I decided to keep it short Okay. So I will start with the basic thing. What is genome? So genome is basically is really a common term nowadays. Nowadays, almost everyone are familiar almost with the genomes term. So life is specified by genome. Every organism, including human, has a genome that contains all the biological information needed to build and maintain a living examples of that organism. Okay. So most of the time, so we do have DNA in it, and we have some discrete unit called genes. Okay, and so there's a brief history about this one. The gene and hereditary unit is actually Johansson in 1909, and then George Mendel come and they start calling them factors. There are many things that you guys already aware of, so I will mostly focus more on informatics section of it. So the basic thing here, the genome and sequence of DNAs, where a genomic sequences and then sequencing is done. So DNA sequencing is actually the process of determining the precise order of nucleotide within a DNA molecules. So this is entire things, okay? The process of determining the precise order of nucleotides within a DNA molecules. So there is PCR with DNPT approaches, and then we have separation, and then we have standard sequencing methods in which we sequence all and their possible order in overall. Okay, here in this one, DNA sequencing, we have a lot more fragments. This is what perhaps it is being already covered by Mukul at some stage. But once we do the sequencing, we have thousands, millions and millions of fragments. And then we do the genome assembly. We assemble all these small tiny reads and form our entire context genomes or scaffold here. So here we need to understand why do we have thousands of fragments when we do any kind of genome sequencing, and then what are the processes that we need to follow to assemble those genomes, those small tiny reads. So remember, I think it is already being covered in previous lecture, but I will repeat it here. So, so if you remember here, once we have this small tiny fragments, and then once we assemble the genome sequences, then when we do the DNA sequencing, we usually have, when I started working with the DNA sequencing reads, at that time, the size of the reads used to be 33 base pair, just 33 base pair. Okay, at that time, 33 base pair was the longest fragment possible that we used to have at that time. But nowadays, we are lucky that we have even bigger fragments that we can use to assemble the genome. And after that, here at this stage in genome assembly steps, at that time we have limited software, but we have a lot more algorithms, new software, new techniques that is being developed by many researchers across the globe, and they are able to assemble the scaffold in a proper fashion. Okay, I will delete this. And so here, overall, the genome assembly is actually is to resolve a puzzle that and rebuild a DNA sequences from its pieces. So it's just like jigsaw puzzle that you played in your childhood. It's same as that. Here we have small tiny fragments, and we have to arrange all the fragments in a proper order so that we can have the entire information revealed. Whereas in the same fashion here in the puzzles, jigsaw puzzle that you play in your childhood, we also have small tiny pieces, and you need to arrange in a proper order so that we can have the entire image of that photos. So in directly or indirectly, we're doing the exactly same thing using the reads. Okay. To resolve a puzzle and rebuild DNA sequences from the pieces, this is what we always do. And in here, we have chromosomes, then we extract the DNA, and then we back sequencing, and the whole genome sequencing, all the reads available, and then you go to the next step and sequence them. Okay, assemble them. Here, there are many standard sequencing steps that we followed over the year, and you remember influenza, Systematic cerevisiae, C. elegans, Arabis thaliana, Homo sapiens, Orygen sativa. This was the time when we were turning. This is the phase when I started working on bioinformatics, and when Arabis thaliana and Homo sapiens were being sequenced at that time in 2000, 2001. And we were excited to know that we do have many more sequencing techniques coming in at that time. And so this was possible at that time using standard sequencing methods, and it was relatively costly, but now it is getting cheaper, and we do have even a lot more genome sequencing done, a lot more genome sequence is being assembled and submitted to NCBI. So here, if you see, at the time, we only had standard sequencing, but now we have the 454, Solexia Illumina, Solid, Iron Torrent, and BB. Okay, so we are perhaps here. So we are perhaps at this stage where we do have a lot more sequencing by PacBio sequencing techniques, and then we have nanopore sequencing as well. So now the time complete sequencing, they used to be very costly, but after all these chemistry improvement, we do have a lot more bigger fragment that can be easily assembled using any computational tools. Okay, let me delete this. So at that time, when people started working with the genome sequencing, at that time they only used to do no sequencing, nothing. They only do one small tiny fragments, and then possibly look for the possible classification at that time. So the complete nucleotide sequences of bacteriophage MS2 RNA, primary and secondary structure of the replicate genes were at that time, there was no sequencing as such, but later on, the whole genome random sequencing and assembly of influenza, at that time, they started working towards genomes assembly overall. And here, you look at this first software at that time, it was Tiger Assembler, and then hardware was just 512 MB of RAM. Okay, so at that time, Spark Center, where you usually have 512 MB of RAM, and there was only one assembler at that time, Tiger Assembler, that they used to assemble this influenza, and they were able to achieve the entire complete genome out of it. So, but the time has changed completely. Tiger Assembler at that time, they only use Smith-Waterman algorithm, and they do all these overlaps check and then assemble in a proper format. And so the things that was not aligned well were discarded at the time. So Tiger Assembler was the initial 1996 first genome assembly program that is being developed at that time. But But the problem at that time was that we only use overlapping methods. So there is two, 3 different methods to assemble. So overlapping is actually time consuming and RAM consuming as well. So, but at that time, the genome that is being sequenced was tiny, and it was possible to assemble using overlapping approaches. But once we turn to move towards Homo sapiens, or let's say Arabidopsis thaliana, that was become even difficult, and we started looking for the alternative options as well. So in previous lectures, Mukul already discussed SRA. So SRA sequence, so fragment treats in separated entries, and then you do the assembly, and then you have shotgun overlapping contigs, and then you all these using overlapping contigs, you have constructed the assembled scaffold or super scaffold in chromosomes. So from SRADs to the contigs, scaffold, super scaffold, and chromosomes that we achieved. At that time, this was the initial protocols that people followed. Nowadays, we still follow the almost same protocols as well to assemble from SRA to scaffold or contigs. And there was a back-to-back sequencing, initial sequencing analysis of the human genome at that time. That is done in International Human Genome Sequencing Consortium, and at the same time, whole genome sequencing, shotgun sequencing approaches being used in the sequences of human genome that is published in Science at that time. At the same time, whole genome shotgun sequencing, when they use the software at that time. So while working with if you look at the Tiger assembler that I mentioned here, the Tiger assembler here, this was not capable to handle the human genome because it's really bulky. So at that time, they started developing a new software, and they developed a new software called the WGA assembler. We call it Kabog. And the hardware at that time was 4 GB RAM was the maximum possible. And all combining in parallel, they had 160 GB RAM and 160 cores in 5 days. So that was at that time 40 machines, alpha SMPs, and this many of RAMs was required at that time. WGA, because this was bulky, human Homo sapiens data is approximately genome size is 3.2 GB, and the reads were even way too bulkier. So we were not able to handle using the traditional old Tiger assembler. So they designed the new assembler that's called WGA assembler, a Kabog, and they assembled using this and published it. The same fashion, there was a separate group working on the sequence of de novo sequences of panda genome, and they used SOAP de novo at that time. So SOAP de novo was the software, and they used Broom graph methodology, and the hardware at that time was 512 GB of RAM and 32 cores. So they used 32 cores and 512 GB RAM, and SOAP de novo was the software that is being used to panda genome assembly. So you see, almost every genome almost have certain complexity, and they have tweaked, customized assemblers as well at that time. So remember here I mentioned Broom graph methodology. So you need to understand what is that as well. So specific n tuples, n-gram or nucleotide sequences of amino acid sequences. So ordered list of element. So n tuple is ordered list of element, and n-gram is the contiguous sequences of n item from a given sequences of text. Okay, so here is the one sequence A with 25 nucleotides, and the 5 kmers or 20 kmers. So if you divide this 25 nucleotide sequences into kmers, 5 kmers or 20 mer overlap, 20 mer length, then we'll have this many of sequences overlapping. So you see, first one is starting from A, second one is starting with T, then G, then C, then here. So you need to understand this one. So because you are learning for the first time, so here you need to understand the first sequence starting from A, and then the second sequences of 20 base pair starting from T, and then the third base pair G, and then next base C, and then G. And all the length is actually 20 mers. So all the lengths are 20 mers, and the last base is 20. So we cannot go beyond this. So 5 mers of 20 mers is possible here. So, most of the time when we do have this mers, small tiny mers, we need to certainly have this overlap. So once we have the short read sequencing of any bacteria, virus, any pathogens, or human plants or anything, we go to we look for the possible overlap between all these, and then using overlaps, we arrange in a specific order, and so that we can have the consensus sequences. So, and then the complexity that we have to check the possible overlap between all these bases. So, and then we need to have Euclidean cycle, and we need to travel only one node at a time, or once, and then we need to form an entire genome out of it. So at that time, this assembly, the Broom graph assembly, become really popular because of the speed, because overlapping approach is really time consuming, whereas Broom graph K-mersbridge approach was easier to assemble genome within an hour. So that's the reason why that people started switching towards different graph approach and including cycle formation and assembling the genome. And then there were many softwares coming at that time. So apart from comparison of two measure classification of algorithm, there is an overlap layout consensus, the first one, and the second one is Dugan graph. So these are the two popular softwares that we had. So the first one is the popular one that people use is Dugan graph here, and the first one that is being already used in the first genome assembly is the overlap layout consensus approach. Is an overlap layout consensus approach. Am I audible to everyone?
Yes,
Yeah,
sir.
hello? Yeah, hello? Hello?
Hello.
Okay.
Yes, sir, you are audible. Yes, sir.
Okay. Thank you. So here, so the two different approaches. The first one is overlap layer consensus that is being already used. So they only look for the overlap between all the reads. Whereas the second approach is the deep-in graph approach, where people are using the deep-in graph approaches to assemble the genome as well. So overlap layer consensus algorithm is more suitable for the low coverage long read, whereas deep-in graph algorithm is more suitable for the high coverage short reads and especially for large genome samples. So if you want to apply OLC approaches is only suitable for small, tiny genomes. So let's say you're going with the viruses or any bacteria that might fits you, and there are many softwares designed especially using OLC to assemble the genomes. Where if you're planning for the long genomes, such as human or plant, then you need to switch towards DBG, where algorithm is more suitable for highly covered short reads, and especially for large genome sequence assemblies. So these two assemblies are popular nowadays, and people using in almost all genome assembly approaches. So now I'm moving towards the sequencing tools and comparables. So overview of sequencing project assembly. So here the FASTQ reads that you have, and then the FASTQ processed. This is the steps where you are actually processing. And then you generate the contigs. Then you do the scaffolding, and then scaffolding is done. We have a scaffold ready for you. Okay. And then you can go to the next step and further go for the super scaffold or chromosomes. So this is overall path that we follow while doing the genome assemblies. Okay. So here the technologies that we have in nowadays and that we use frequently, that is here Illumina is the one that most of the time people use. Just wait. So here the Illumina, this is the one HiSec and 10 is actually they approximately 3 days to generate 2 into 15 base pair, and the total number of reads generated in 3 days is this much, and approximately 1,600 GB data is being generated. Okay. Here, Illumina MySec, it took only 65 hours, and then approximately 13 GB of the data is being generated out of it. And then Solid and IronTorrent and PacBio. And PacBio takes only 1.5 hours, is a long read assembler, and then generate approximately 0.3 GB of data out of it. Okay. So the benefit that all these techniques that have is some of the read size is smaller. So here, if you see here in this Illumina HiSec, we have 2 into 150 base pair, whereas Illumina MySec, we have 2 into 300 base pair. So that is even bigger reads that we can use for any genome assembly. And Solid, if you're using 2 into 60 base pair, and then IronTorrent is 200 base pair only. And then PacBio is approximately 850 base pair. It's a longer fragment as compared with other short sequencing techniques. So we can opt as per our need and the money as well, because sometimes if you go with IronTorrent, it's much cheaper. It takes less time and generate a lot more reads, and it's cheaper as well. So you can go ahead with this IronTorrent as per your need. And most of the time nowadays, I prefer myself Illumina MySec, because it's a 302 base pair, and it generate two. Its quality is really good, so that's the reason that we started following, and the techniques that we mostly rely on is Illumina. And whereas PacBio might be possible, they are generating bigger fragments, but the problem with this quality, and we might need to filter sometimes some of the reads. And I personally do not prefer PacBio over Illumina. Okay, so there are many other technologies as well. So apart from this one, so here is 454 parallel sequencing, and in here the read length is approximately 450 base pairs, whereas Illumina HiSeq is 600 GB sequence yield and low error rate. So Illumina HiSeq is also preferred. MiSeq is 250, and solid is two base encoding reduced the observed raw error rate, as ion is fast run and PacBio is long, approximately 3,000 base pair. So it does have weaknesses. All these techniques have some weaknesses, and we certainly need to plan our experiment accordingly, or request for any sequencing techniques accordingly. And so you can go ahead with this one. I will share my slide to you as well for future references. So libraries type, so orientation, single reads, forward, just forward reads most of the time being used. Pair and 500 to 800 base pair insert size, and then we have forward and reverse, exactly same sequence in forward and reverse order. So Illumina and mid pair, where we're using 2 to 40 KB insert size, and we have a reverse forward and reverse, and so both Illumina and 454. So the length varies, and that can be useful for future analysis as well. So why it is important to have a pair information? So if you look at the de novo assembly, all the reads overlaps, and we can create a consensus context. Whereas if we have the pair end information, so forward and reverse, and in between we have n and n, we can use this information. We have longer read information that we can use to scaffold or supercontig formation as well. And genetic information, so if they have the specific markers, we can use this marker information to form an ultra context. So these are actually important when someone is doing any kind of genome assembly analysis. So sequencing amount, so ingredient for good assembly. So most of the time when we see that someone need to do this genome sequencing or assembly, we usually request for the good amount of read coverage and read depth. So read length also matters for us. For example, if someone is doing the read sequencing, I always, as a bioinformatician, we always request for longer reads. So longer the read fragment, better the genome quality. This is always we believe that, but it's not always true. But sometimes, even if you go with the PacBio, but PacBio have a lot more error rate, and that will be difficult for us to handle in later stage. So sometimes we go ahead and we'll simply say that best quality reads is needed, but read length always matters for us, especially to resolve the repeated region in the genome. Coverage also matters. So let's say once we are doing any kind of sequencing and requesting for 10x, means 10 times being sequenced, one region is being sequenced 10 times, and then that will be not more than enough to resolve some of the repetitive region in the genome. And for that one, we request them for have 40x or 100x sometime, or sometime in viruses case in COVID-19 virus assembly, we requested 1,000 times x as well. So that will help us to reliably call a specific mutation in the genome. So the coverage matters as well. And the quality, obviously, if the quality is bad, then we simply discard all the reads. And we always need good quality reads to assemble and do any kind of analysis. So all these three things really matters for us: read length, the coverage, and the quality. If all these are okay with us, then we go to the next step and do the genome assembly and do all sort of analysis. Depending on the genome's complexity, technology used in assembly, so how many sequencing amounts you need. So more is better always if you have enough computational resources. So always say like we need better read length, better quality, and we need to have better computational power as well to do any kind of sequencing assemblies. So more is better always. But Sanger 10x less than 10 is not good approach, whereas 454 20x and Illumina 100x is required. So more than 100x data that you have, you can simply go ahead. Sanger 10x, Sanger is 100% correct. So you can we can rely and we can even go ahead with 10x as well. But Illumina 100x is always recommended by us, and we always request the users to sequence at least 100x so that we can have the complete assembly done in proper with good reliability. Whereas in polyploid or higher increased amount of reads are needed. So if you have the high ploidy level, then you need even more than 100x because sometimes you need to separate these ploidies as well. And if the genome is highly heterozygous, then also need good amount of reads to understand this ploidiness and heterozygosity in the genome. If someone is dealing with the plant genome, for example, where we have hexaploidy, so at that time, in that cases, we need even higher X number of genome coverage. The use of different libraries types. So we also need pair and met pair information as well to assemble it properly. And the longer read is preferred. And if we have longer reads as well while doing any kind of genome assembly is highly preferred. We mostly use the longer reads to assemble the genome and scaffold at the stage of scaffolding or creating a super scaffold or chromosomes. We use longer reads. So assembly, so we usually opt for the hybrid assembly where we do have shorter reads, longer reads, and then we assemble using all these shorter and longer reads to assemble the genome. So here, this is the complete flow where the FASTQ raw reads, FASTQ process, and the context formation, scaffold, and finally the chromosomes. And we certainly need to make many decisions during the assembly. And for that one, you hopefully Mukul will discuss you and the entire assembly flow, and he will help you to assemble one genome as well. So FASTQ, you see, he is already discussing with you that we have fast QC, that we can do the QC of your reads that you have. And then you can go ahead with the training. So we have FastX, there has been SEC, there are many other tools that you can, Informatics, and many other tools as well that we can use to train the sequences. And we also need to remove sometimes the contamination in the genome and in the reads. So if your reads file does have the contamination, then you need to remove those contamination before doing any kind of assembly. So in that case, you need to decontaminate your samples as well. So for read correction, so there are many approaches that people follow to correct the reads. And so there are softwares that you can use to correct the genome assembly and create a consensus out of it. So the read correction, so there are a software that we call it Reptile or Eco or Corrector, Quack. And there are many softwares that are available that can be used to correct your reads before doing any assembly. So this is always not recommended. Sometimes some of the software might insert some random mutation in your reads. So that's the reason why that people sometimes avoid doing the correction, and they always use the raw reads. They prefer to use the raw reads and doing the correction at the end. Sometimes people prefer to do the correction at the read level initially and then genome assembly. So is always up to you, but I prefer not to interfere with the read itself in the very initial stage and do the assembly and then check for the correctness at the end after the genome assembly is being done. So it's just up to you to opt. And then we go ahead with the genome assembly softwares. Though, as I mentioned earlier, there was Coboog, MIDA, Alpha, LG, Soap de Novo, Velvet, Ray. There are many software that is nowadays outdated. No one is using that software nowadays. But I used during my journey, during my assembly journey, I strongly recommend MIDA because I myself tried MIDA and it works really well. It actually works on an overlap layout consensus approach algorithm, and it is really designed well for bacterial genomes. So if you are dealing with any bacterial genomes, I will recommend you to try Myra at least once. So it does have very good quality and it does have very good accuracy as well in genome assembly. Soft de novo was the popular at that time, but nowadays is being outdated. There are many new softwares and coming in that is better than Soft de novo, and Velvet as well. You can try. So Ray, Velvet, and Soft de novo that you can try to assemble your genome if it is needed. There are many other softwares. Mukul will cover that one. The new softwares to you. So all the softwares have some weaknesses and the benefits. For example, if you try with the SOAP de novo, is the fastest. So it's really quick. It can assemble your genome, but accuracy is concerned sometimes with SOAP de novo. So you need to handle carefully. Whereas if you try MIRA using, let's say, long genome, it will create a lot more problem because it is really slow. So you need to opt according to your to your need. So there are many assemblyism being organized by many communities to further look for the critical evaluation of genome assemblies and assembly algorithm. So, and they were actually having, every softwares have some benefits, some unique features. So it's just up to you to opt. So as per your need, if you think that you are dealing with the bacterial genome length is really small and the number of contexts is sufficient enough to do the MIRA assembly, then you can go ahead and do the MIRA assembly by yourself. Then, okay, once we have the assembly, initial assembly done, then we have the contigs. And sometimes you need to fix this NNN in the genome. So there are many places in your genome assembly that you might have NNN inserts, N's inserts in it. And at that time, you need to fill this gap using gap closer, gap filler, or PB suite. You can use any of this or many other softwares as well that can help you to fill these gaps. And if once this gap is filled, it will have a proper genome assembly with detailed annotation possible. If there are any gaps in between, then there is a problem with this annotation software, so they might discard those regions completely. So these softwares are freely available, and you can try by yourself as well. So there are many computers. Which kind of computers we need to do any kind of genome assembly. So the computers, the bigger is better. So how much do you need is depends upon your genome, right? So if you are actually doing human genome size assembly, then you require approximately 256 GB to 1 TB size of your specific RAM. So how many reads do you have? So it also matters first. So let's say you are having 300 GB of data. So you certainly need a specific hard disk to store all these reads. And you certainly need to have this much of RAM so that you can process it in a proper way. So you also need to have a good amount of RAM, good amount of storage. If you're using OLC approaches, then you're going to keep all these reads in your RAM for any kind of processing. And you to check for the all against all overlap, it will take a lot more memory than DBG. And many parameters are going to use for you. So bigger kmer, more memory. So if you're using kmer 21 versus kmer 71, then you need more memory for any kind of analysis. Uh, so it's up to you, but it is always recommended bigger the better. So if you have bigger assembly storage, better. If you have bigger memory, better. Bigger read length, much better. Bigger kmer size, much better. But it all have some limitation as well. And so it is up to you to decide which one you want to opt. There are many options available nowadays. Nowadays, it's possible to have 1 TB of RAM, and you can spend good amount of money on it. So options available nowadays in the market. And so when we started working with the genome assembly, 1 GB or 4 GB were even the maximum possible. But nowadays, is 1 TB is normal. So you can opt nowadays and do the genome assembly by yourself. Now it's coming once your genome assembly is done. How to assemble, evaluate those assemblies is always concerned. So how far is this value from its estimated genome assembly? So you need to estimate your genome size first. So let's say your genome size is 3.2 GB, then you should know and you should aware to further validate it to the number of context scaffolds. What is the N50 of your genome assembly that we need to look for? The longest scaffold in context always matters. So we have, let's say, chromosome scale assembly, then it's much better. Average and context length and N50, all these need to be calculated and used to evaluate your genome quality overall. So here is the N50. So sequencing, so you need to arrange all these reads in this proper order. The places where you achieve the 50% of the reads is the N50, and you can use that threshold to report to any journals to accept your genome assembly. So hopefully Mukul will discuss you with N50 and L50 in detail with command line. So assembly evaluation. So once we do any kind of genome assembly evaluation, you always have genome size, the context, the scaffold, and the N50, L50, KB, longest, smallest, and total genome size. That all these stats matters at the time of genome assembly evaluation. So you need to look into all these numbers carefully. Okay, so assembly stages. Assembly stages. So where so we do have like complete for non-repetitive regions, complete for non-repetitive regions, complete for non-repetitive regions, and then complete for almost all whole genome and the whole complete genome. So usually from a small tiny scaffold and fragmented, then we have a small medium scaffold, then large scaffold, then the pseudomolecules and the so all these matters, when someone do all these genome assembly, we need to certainly improve over next every run to fix the genome assembly and achieve the chromosome scale. So ultimate goal is to achieve the chromosome scale assembly. So I will skip this one. This is not useful. Okay, so here, once we look for the complete repetitive regions and genome sequences, we certainly use jellyfish. So sequencing data, kmer count. So you can opt for the especially jellyfish or FC counters to look for all these sequencing kmer counts in your genome, and then we can use these kmer counts to estimate the possible sequencing assembly completeness as well, to check for the completeness of the genome and there's the genome size. Estimated of genome size, you can estimate the possible genome size and the assembly size that you achieve. And then you can look for the possible genome not assembled, so 150 to 120. So here, the estimated kmer count, say 1.38 GB, should be the length of the genome, whereas assembly size, say, is 1.26. It means we have certainly missing part in the genome assembly that you need to fix to achieve the best quality assembly. So the last section of this entire presentation is moving towards the things that you should know about genome assembly. So they have variable size. For example, an angiosperm plant, they range from 63 MB to 150 GB with an average of 5.6 GB. Okay, so the genomes are of variable length. So sometimes the genome length is 63 MB, just 63 MB, whereas sometimes it goes to 150 MB. So these numbers, even in plant and genomes, the genome size is variable, and you need to know first before you do any kind of genome assembly. So it is recommended to estimate the genome size before you do the genome assembly, so that you will have the basic idea that we are speculating to have at least this many MB or GB of the genomes that you should achieve. They can be polyploidy. So if genomes are polyploid, that is even more tedious. This is the reason why that most of the time the people say that plant genome assembly is much more tedious than basic diploid human genome assembly, because human genome has two diploid copies, whereas polyploids are even way too more trickier and required more skills, because we need to deal with this homozygous and heterozygous region and the collapse region in the genome assembly, that we need to carefully deal with this. Highly heterozygous and polymorphic are always a problem. So homozygous will collapse them completely. Heterozygous will not assemble at all, and they will have a highly fragmented genome assemblies that we certainly need to take care in detail before we do any kind of genome assembly. There have been recent whole genome duplication events and collapse during the assembly. So this is a big problem when we do any kind of genome assembly. So genome assembly have whole genome duplication sometime, or let's say some certain genome duplication sometime, and once we're using any software, sometimes we forcefully merge them into one genes. So multiple genes into one genes, or sometimes entire genome is duplicated and we decided to merge them and form one single genome. That is not good, and we certainly need to look for all these before we do any kind of genome assembly. Repeats in the genome is the nightmare for us. So most of the time, once we have highly repeated genomes, regions in the genome, then it is really difficult to assemble them, because sometimes always repeated genomes are hard to resolve. And for that reason, we always request for we request for the long read assemblies. So to resolve these repetitive blocks. So this is the reason why that I prefer myself to have some short read sequencing done than a long read, just to resolve these schemers, these repeat region in the genome. So hybrid assembly approaches works here, and so you need to decide by yourself which one you want to assemble, and if it is highly repetitive, does it have a highly ploidy level? Then you need to request the reads as per the need, as per your requirement. Collapsing problem is the bigger problem in genome assembly. So let's say you do have exactly gene A1 and gene 1B, and all collapse into one and form one gene, which is bad idea. So it is better to not collapse the gene regions, and if they have a high heterozygosity or low heterozygosity, then you need to handle it carefully before you do any kind of genome assembly. So highly recommended, do not collapse your ploidy. So if you collapse the whole genome duplication events, then there will be a problem, and you may misclassify your entire plant genome as well. So it is recommended, highly recommended, please handle your duplication, whole genome duplication, even gene duplication carefully before you do any kind of genome assembly. This is the reason why that we always request from physical map data. While we're doing assembly, we sometimes request for physical map data to understand the duplication level in the genome as well. So how do you actually evaluate the collapsing fragments? So I personally prefer the Kimmer approach to validate the collapsing scenarios, where if it is collapsing, they certainly have the missing k-mers in this read file or assembled contigs. And if it is not there, it means it might be possible it is being collapsed during the genome assembly, and we need to handle it carefully. There are many other approaches that people follow to assess the genome assembly, and there are many software that is being designed to quickly assess the genome assembly, to easy to apply for the current technologies. And there are many software that is being developed by many bioinformaticians. The Minimus is the one that I use by myself, and to look for the existing integration of big sequences in Minimus do as well. So you can try one of them, but yes, if you are going ahead with doing any kind of genome assembly, be careful. Do not collapse the context or reads. As I mentioned earlier as well, repeats are the nightmare for us, and so you first need to repeat mask the reads, or sometimes you use the repeat explorer to look for the possible repeats in the genome, and then fix it before doing any kind of an assembly analysis. So it is highly recommended to decontaminize yourself and look for the repeats and resolve before doing any kind of analysis using those genomes, assembled genomes. That's all from my side. Thank you very much. And if you have any question, let me know. Sorry, it's being one-sided.